TK the Science Guy.
TK the Science Guy.
TK the Science Guy.
APES rule.
TK the Science Guy.
Wind power. I'm a big fan.
Time.
TK the Science Guy.
TK the Science Guy.
Hey CW Apes, Mr. Kennedy here. Today we're going to do an experiment called the Allelopathy Lab. That's a big word that really just describes, well, for lack of a better way to put it, kind of like some chemical warfare that plants wage on each other. Have you ever walked around outside and as you see different plants, different trees, noticed that in some cases there's nothing growing underneath them, but others, grass and other small plants, can grow right up to the trunk of the tree? That's what allelopathy is all about. Plants can literally wage chemical warfare on each other to prevent other plants from growing underneath them. You might ask why? Well, it's all about competition for resources. So that's the experiment that we're going to do today. You can find the handout for this experiment on our class webpage under the Living World Unit 3. This is what it looks like. The Allelopathy Lab. So the first thing that I'm going to do for you is show you some of the materials that I'm going to use to test allelopathy. First thing that I'm going to use are 6 Ziploc bags. That way I can establish a control and I can have 5 other test conditions for my experiment. Second thing I need, some seeds to grow. Now, if you look closely, that's a big cup of bird seed. All kinds of different types of seeds are in this bird seed, so I guess I have to sort them out. That doesn't look like fun. Whew, that was a lot of seed sorting. Now that I have the seeds all sorted out, there's some other stuff that I'm gonna need. I need some paper towels to hold the moisture inside my Ziploc bags while my seeds germinate, or not, depending upon the chemistry of the plants that I've collected. So I went outside and I noticed there's these trees outside of my classroom that have these lovely green leaves. And wouldn't you know it, nothing's growing underneath them. So I thought, well, that would be a great candidate to use for my allelopathy experiment. What I now need to do is to grind these leaves up so that I can extract the juice, right, or the chemicals from them to see what impact I'll have on my seed germination. Off to the blender. Okay, gang, I'm over here at the blender. So I've basically taken all of my leaves and smushed them into the carafe for the blender. We're gonna mix them up and make a milkshake. Looks yummy. All right, here we go. All right, the blending is done. Doesn't it look yummy? Yummy. Okay, gang. Now it's time to set this experiment up. Now this is a controlled experiment, and if you remember all the parts of a proper experiment, that means we have to have a control group that we do nothing to, and an experimental group that we apply our variable to. So in this case, we're gonna look at the allelopathic effects of our green milkshake on seed germination, specifically red millet, which is what birdseed's made out of more than anything else. So as I set up my Ziploc bags, each bag has to be treated exactly the same way with the exception of the liquid that I put in it. So I'm gonna start off by taking some of these paper towels and I'm gonna put them in each one of these bags, right? That way the seeds have something to hold the moisture in. See what that looks like? all 6 of my Ziploc bags are gonna get paper towels. Just like that. All right, guys, all the paper towels are in the bags. So the next step is for me to label the bags so they know what's in them. So I'm gonna put like control on the one that's my control, and then the other 5 bags, I'm gonna label with some quantity of the green mixture that I'm gonna put in there on my seeds. And I say some quantity because, well, it's a controlled experiment and I don't wanna just pour this stuff in without knowing how much I put in there. So maybe I'll do it in mils. Like maybe I'll say 5 mils for my first bag, right? And then go up in increments of 5. So I'll have a 10 mil bag, a 15 mil bag, right? 10 mil bag, a 15 mil bag, and so on. Once I have that all set up, the next step is to put my seeds in the bag, and finally, to fill the bags with the appropriate liquids so that we can let nature take its course. Okay, gang, so here's my bags, all properly labeled. Now it's time to fill them up with all these seeds that Mr. Kennedy had to sort out a little while ago. Okay, so as you can see, each bag now has an equal number of seeds in it. So all that's left to do is to add the liquid and wait to see what happens. All right, gang, so there they are, all finished. The control has 5 seeds, so does the 5 mil, 10 mil, 15 mil, 20, and 25 mil, all set up. They also have been watered. The control got plain water, the other bags got increasing concentrations of plant mixture. Now remember, that plant mixture is what we're using to look for allelopathy. So there's nothing left to do now but wait a few days and see how it turns out. Hey CW Apes, Mr. Kennedy here. It's been a couple of days and it's time to wrap up the Allelopathy Lab. So I set this up with you a few days ago, walked you through the handout and what it looked like and where to find it. So it's all about collecting results at this point. Let me show you what happened. As I show you each of the bags that we set up about 72 hours ago, your job will be to count how many seeds actually sprouted after being exposed to that liquid mixture that we produced by grinding up leaves from the tree just outside of my classroom. That's our data. If there was an allelopathic effect, well, I would assume the seeds wouldn't sprout, wouldn't you? If all the seeds sprouted, then that tells you that, well, whatever chemicals were in the leaves had no effect on the seeds. Check this out. This is our first bag. It's the control. And remember, the 5 seeds in this bag were exposed to nothing but plain water. So as I zoom in, I want you to count how many of the seeds have actually sprouted. How do you know if they've sprouted? Well, something should be sticking out of them, like the formation of an early root. So count up how many have sprouted in the control. This is the second bag. Remember, it had 5 mils of the plant leaf mixture that was all ground up, mixed with some water. And as we zoom in, you're gonna look again to see how many of the 5 seeds have sprouted. How do we know they've sprouted? Well, something should be, well, sticking out of the seed. This is our third bag, 10 mils of the leaf mixture and water. As you look closely again, you need to count how many seeds have sprouted. At this point, we're starting to get into the higher concentrations of leaf mixture. And as you look at this bag, this is our 15 mil bag. Look closely at the seeds and count how many have sprouted. This is our 20 ml bag. Again, how many seeds have sprouted? This is our last bag, the 25 ml bag. We're looking again for any seed germination. Be careful not to mistake a glare on the plastic bag for something coming out of the seed. So that's the end of the Allelopathy Lab. You've now seen the outcome of exposing red millet seeds to a mixture of ground up leaves from a redbud tree that sits just outside our classroom and water. Use that information to interpret whether or not allelopathy occurred in these bags. This data should also be used to answer the other questions in your lab report. As you look at the questions in the lab report, refer back to the video, rewind it, play it again, look at how the experiment was set up. What were the working parts? What were the control groups? The experimental group? What was my variable? And ultimately, what was my data? Don't forget to include things like, what was my hypothesis? And then conclude. Was there an allelopathic effect? Did the redbud leaf mixture have any impact on seed germination? If so, how do you know? That's what this experience is all about. I hope you've learned a little bit about allelopathy, that chemical warfare that plants wage on each other to, well, outcompete their neighbor. I hope you've enjoyed this. I'm Mr. Kennedy, and don't forget, reduce, reuse, recycle. Keep it green, apes. See you next time.
TK the Science Guy.
TK the Science Guy.
TK the Science Guy.
APES Rule.
TK the Science Guy.
Wind power. I'm a big fan. TK the Science Guy.
TK the Science Guy.